Hi, I'm Chris Chadwick, Vice President of Healthcare Services here at MAC. Welcome to the next in our series of video podcasts. This time we're going to be exploring the acceleration of early clinical development. Before I introduce all of my guests, I'd like to remind you that MAC Clinical Research is a global full-service CRO with science at its core. With a fully owned network of high-performing clinical sites and many specialist facilities, such as sleep laboratories, our first-in-human MHRA-accredited early phase unit, psychedelic suites, GMP manufacturing and pharmacies, as well as state-of-the-art laboratories. Today, I'm pleased to say that I'm joined by three guests, Dr. John Connell, Teresa Cullen, and Jorge Costa. Welcome. Collectively, they take responsibility for all scientific aspects of our early clinical development. And I'd like to initially ask each of them if they could tell us about themselves and their role within MAC. So, John, I'd like to start with you if I can. And could I ask you if you could also cover the function of your team and what they do at MAC?
Yeah, happy to, Chris. I suppose my career started about 30 years ago. I've always worked in early clinical development. I've worked for most of the large early phase units in the UK and over in the United States. During that period, my drive was always to find ways that we can develop drugs faster and much more sensibly to get drugs to market faster and reduce the patient burden. About 10 years ago, I got the opportunity of joining MAC, where I'm now Chief Scientific Officer. The reason why I joined MAC was I'd worked with them previously. They have 10 sites, I think you've already mentioned, which were recruiting fantastically into studies. It gave me the opportunity of joining at a time when we were opening an MHRA-accredited Phase 1 early phase unit, so we could do first in mans. We could combine that with the patient work. But more importantly, in my career to date, I spent a lot of time developing techniques and using models of disease, which were there to accelerate development. And to do that, you need a strong scientific base. And that's where these guys come in.
Okay, thanks, John. George, would you like to introduce yourself?
Yes. Hi, Chris. Thank you for having me. I'm Jorge Costa, and I've been with MAC for the last 6 years. I have over 12 years of experience in neurophysiology and sleep assessments from NHS. Here in MAC, I've been leading the scientific solutions, which is basically like where we specialize in two of the techniques. One is the clinical pharmacodynamics, and the other one is the clinical rating scales, where we have specialists with a loads of expertise in these techniques. In MAC, we cover from phase 1 to phase 4 studies, which can be like from healthy volunteers to real patients. And I'd like now to pass to Teresa so she can tell a little bit more in detail about this.
Perfect. Thank you, George. Teresa.
Lovely. Thank you. My name's Teresa Cullen. My background's neuropsychology. So I work with John and George within the Scientific Solutions department. My specific role within the department is Clinical Science Manager. And that involves me leading the scientific team as one of the branches of Scientific Solutions. Part of that is adding any of the particular scientific measures to the studies and leading the team to come up with the ideas, work with sponsors and develop these alongside them, as well as also I'm one of the core raters within the business. So this involves me working with the rater team because of my psychology background. It means I can implement these sort of gold standard measures throughout any sort of particular clinical trials, which is with depression, sort of anxiety, PTSD, and also one of the new trends, which is the psychedelics, which is coming through the market.
Perfect, thank you. So leading on from that, you touched upon specifically about different techniques, and I just want to see how they can advance clinical development. John, can I have your thoughts?
It's a great question, Chris. Obviously, there's a lot of experience that we've got, so to pick from is difficult. One of the examples I always actually give, though, is many years ago I was actually working over in the US. We were working with a client who was a very small biotech, and they were really struggling financially at the time. They basically had a great compound coming through, which was a novel mechanism of action. They just about had enough money to do a phase 1 first-in-human study. And from there, they were going to try to get funding to carry on the development. The study itself would have wiped them out completely. And we had a good chat about how to design the study. And what I realized was, as this was a drug which was affecting the opiate system, perhaps we could put additional measures into that study which would not inflict in any way with the safety, the tolerability, the PK, which they had to go, but would give some indication they were actually hitting mechanism and, give them some indication they should really push forward with that drug. And so we managed to incorporate into their study some measures of pupil activity. We used pupil diameter measurements because if you attack the mu-opioid receptor, you get a decrease in pupil diameter. Because the drug was being developed for pain, we also put one of our pain models in, which we validated and spent a lot of time working on. To cut a long story short, they got their safety, they got their tolerability, they got their PK data, but they also got the perfect PK-PD match on this change in pupil diameter. And they got a signal that they were reducing pain because the pain response in what was a very simple cold pressor test was actually also reduced. Now, that gave them the confidence to carry on, but more importantly, they then went to investors and showed them that data. And now investors see probably thousands of compounds a day where there's good preclinical data, there's a little bit of safety data, but to see the fact that they were hitting the correct target made a massive difference and increased their funding, and they were able to take that drug forward by including those particular measures. So that made a very big difference to them.
That's fantastic. Thank you. So, Teresa, any examples you can think of you'd like to share with us?
So we worked on many different types of compounds, as John was explaining. And one that probably springs to mind is one that myself and George worked on, which was the capsaicin model. This was a compound which we did a four-way crossover study within 2 months, and so it accelerated the research within that area. Have you got anything else you want to?
No, it was very interesting for me to use these different techniques because we mimic, of course, symptoms like the capsaicin model is used like to mimic a pain model. So we would see like neuropathic patients, and this way we can actually get into the healthy volunteers and mimic these symptoms and actually measure in a quality way how that would be translated to the drug development.
Yeah, a good example of one actually, because I remember traditionally we'd done a lot of work with neuropathic pain studies going straight into patients, and those studies could take a year and a half because you got to recruit the patients, you got to perhaps take them off their medication. Did you finish that in about 2 months?
2 months.
Yes.
Then you got like a month or so for number crunching, but you can quickly see that the drug that we were testing reduced hyperalgesia, our dynia was reduced. And that's what you're looking for in those first pain studies. But of course, you're moving it forward 12 months by doing that.
And that's phenomenal to be able to cut that timescale by so much.
Yeah.
Because we have a small and dedicated team that specializes in these techniques, it makes it a lot easier because we can be mobile and going to these sites and conduct the work from there. Like another example that I would like to say to you is from my own experience because I have worked as a clinical neurophysiologist. I used to see like my tests, my assessments only for diagnostic, and I ended up like using these assessments also for research. Like one of the studies that we have conducted a few years ago, it was with toxins, where we're comparing 3 different toxins. Everyone knows about toxins because or using for cosmetic reasons, like for the wrinkles, or for conditions like migraines or other ones. I think not like about toxins, but we wouldn't know like, can you do this assessment for this specific case? And we actually compared these 3 toxins like in a very good way, like and how we done it. Like we used EDB model. What this stands for is extensor digitorum brevis, which is a small muscle in your foot, basically. And why we got it like in your foot, a toxin, because it's easily accessible and it makes it easier as well to measure. So basically, we have conducted like nerve conduction studies, which is electrical stimulation. We'll see how the the nerve and the muscle will excite, basically. So you will do this electrical stimulation, and this will have a reaction. So we want to see if that reaction, like what is the onset latency, like what is the amplitude. And after you injecting this toxin, we can actually have a comparison study, because even they being very similar in terms of working wise, like it can be different. Like when you have a painkiller, you have different types, like ones that will work very quickly and other ones will be more prolonged. With this technique, we could compare these three well-known toxins in a very good way to collect this data. So it was really fun for me to work in a different way than I used to do.
That was a long duration study, wasn't it? That was going on for over a year. You're measuring the...
Yes, that was pretty impressive. Like
it was 40 patients over a year, which we kept all 40 patients. We had no dropout rate within that patient population. So we... very good study.
No, that's perfect. Thank you very much.
Can I throw a caveat in? Because we give them three great examples of how it always works. But let's be honest, it doesn't always work out as you expect as well.
Yes.
If you reminded me then we were talking. One of my original studies were actually done as part of my PhD, unfortunately, where we took 120 people and put them through anxiety models. Now, anxiety models are to induce anxiety in somebody is difficult. What's really good is actually put cameras at people and make different. It's like that help very nice, which is lovely. And we adapted that. We actually used a simulated public speaking model. And we also did some conditioned anxiety as well. But 120 people were put through both these models over a period of about 4 months. It's a big, it's a really big undertaking. And at the end of it, we were not able to separate any of the treatments we gave people against placebo. People were as anxious on the treatment as not. When you tell a drug company that their drug hasn't worked in one of your models, the first thing they want to do is actually sort of say, well, you've done something wrong, clearly. You should have used a positive control, which, by the way, I agree. If you should have a positive control in these models, but we didn't. So it didn't work. And what they did was to ignore the data, and they ploughed on into patients, and the drug failed in patients. Now, this was obviously a while ago, it was 25 years ago, and people were racing ahead to get drugs into man quickly at that stage. And the reason it actually failed was the drug didn't cross the blood-brain barrier. It didn't go anywhere. It was basically being swallowed and passing straight through. Right. It could not affect anxiety, and it shouldn't. Drug development was the wrong way around at that point in time. They were not checking, in humans, does it get into the brain? But more importantly, they didn't listen to the signals of early phase. They had a perfect opportunity to look at the data and to stop that development of that compound, but instead they ploughed on and gave unnecessary exposures to patients. And it's quite interesting. It was an expensive validation of my model, which I wrote about extensively. That was quite nice. But it just showed me, I think, a very important thing, that you need to make sure you get the steps of drug development correctly. You can't assume what you see in animals, you're going to see in patients. And that's why I think a lot of our techniques, whether it's the electrophysiology with the muscles or whatever, or even any of these models, it's the translation that is really important, that we're trying to bridge that gap between uncertainty in preclinical models and definite efficacy down the line. And that's where we are contracting timelines and making a difference, I believe.
Yeah, we have another study that we've worked on, which is we implemented a food model, and this was a dose escalation. It was a PK. If you originally could just be kept as a PK study to see when the drug is within the blood system, as a lot of studies do. But actually, this particular study, the indication wasn't seen until much later on. But the food model, because we implemented this food model, we actually could see it a lot quicker that the drug was taking effect. So by adding these scientific measures, it really adds value to the...
Yeah. We saw effects on that model, the concentrations where you couldn't even see it in the blood, the drug, but it was affecting people's appetite. They were getting a controlled stimulus where they had a certain amount to eat, and they were eating less. That was amazing to actually see that before the blood told you it was
much earlier on in the study. So
yeah, I can see. Well, thank you. Some great examples there, which I think are wonderful. But I want to move on and ask some other specific questions, if I possibly can, whilst I've got you all here. Just in terms of timings and other added benefits, such as cost savings and indeed removing patient burden, which is so important. What is the impact when considering these, and how quick can studies truly be accelerated?
That's a good question. I think what you need to remember is that there's some of the early phase studies have to be done anyway. You need to do food effect studies, you need to do SAD studies, single ascending dose, multiple ascending dose studies have to be done anyway. So what we're talking about really is adding techniques into them. So immediately you're not extending timelines. All you're doing is collecting more data. Now, it's clearly important that when you put these techniques in, what you're not doing is in any way compromising the main aspects of the study. They have to still be collected. And so in terms of timeline effect, the direct effect is minimal. Where the saving comes in is if you can include in your first administration some indication, like the example you gave with the food effect model, where you're hitting a target, it can give you so much more confidence. It can tell you something about the dose that you should perhaps be using in your next studies. I think without the food data, we'd have gone a much higher dose into the next study because we'd have waited to see an effect in the blood. But we know it was already working, even though it was not detectable. And that's a really important example that we can give. What companies want to do is to get to market as quickly as possible. And anything we can do to shorten that will help people in the long run. One of the best examples, I think, is a study we did about 5 years ago now, a dermatology compound, where in our MHRA-accredited phase 1 unit, we dosed a drug for the first time. We gave it to a healthy volunteer. We made sure it was safe, it was well tolerated. Then the treatment, because it's now going into a patient, was given to a patient for the first time, and that was also done in our unit in Manchester, a phase 1 unit. We then took it into the community to our outer centres where we do a lot more late phase work, and we're able to show that when they took this home and used it in a more normal environment, it still worked. Now, what we're talking there potentially is three studies. We could take 5 or 6 years, but in fact, we did it in a single protocol. We dosed the healthy volunteers, we dosed the patients, we did the community work in 8 months. Phase 1 to the end of phase 2, that probably knocked about 2.5 years off the development time.
Wow, that's really
and that's where we get excited.
Yeah, I can see that. Yeah.
Another example that we have done like recently when John and Teresa was saying is like, of course, from preclinical data, we can get like valuable information. We have done a study that we worked very close, first of all, with sponsors, and we tried to help them adding value to their drug. And in this situation, we were conducting a study for sexual dysfunction, but the preclinical data was showing that it could also be used for pain. So what we conducted in parallel, it was a study for pain, where we done the capsaicin model. Capsaicin model is something that actually hurts a little bit, let's say.
It's injected intradermally, it's painful.
So we have tried, but again, what we were trying to see is like mimic the neuropathic pain. So we could actually show by measuring in healthy volunteers how they would react for the pain and using that particular drug. And what we noticed with that study is like we could reduce in around 7 months this just by proving like with our design that we would go for the endpoints that we are looking for, and ended up like targeting the right biomarkers.
That's a great example because we did that in parallel with the sexual dysfunction study. So that study was running anyway, and nothing could happen until after that study had finished. But in the meantime, they got this secondary indication, so that if the first indication fails, you got a backup, which is great, particularly for smaller companies. I mean, what I would say is that the examples we tended to give so far are for smaller biotechs. But for the bigger companies, the big players in the fields, they've often got a range of compounds that they want to bring through. They may have one, a lead compound, backup compounds. We can actually use some of these techniques to help the bigger companies in screening, and that's something we need to bear in mind. It's not just a technique which is there so that smaller companies can get extra finance, which they often do. This is the main reason people do this is for finance. But for the bigger companies, they can screen and really help them select which are the best compounds to move forward quickly with.
And I think that's invaluable to have that level of flexibility. So, yeah, really, really good. So I just wanted to see, picking up on some of the answers you were saying there, are there any particular therapeutic areas at all or indications that are more likely to benefit from this?
I mean, there's so many, as we can give you many examples as we already have. So we go into lots of different areas. So our team, which as we said is sort of a dedicated team, which is fostered with the two branches that are from the psychedelic clinical raters and then also the pharmacodynamic side, is that we can cross over to many different conditions. So from this sort of the dermatitis to sexual dysfunction, we can also look at things like anxiety, depression, PTSD, OCD. And with the scientific team, so whether it's clinical rating, we'll implement these areas, or we can add them, one of the many models that we come up with. But we also not just use the old things that is kind of already tried and tested, is we'll come up with new ideas within the business as well, and within clinical research, and we work with those sponsors to see what their compound is and how we can add value to their drug by them telling us the preclinical data indications, and kind of then we can come up with them with ideas, research, train either with their experts or come up with our own ideas as well.
Yeah, like the examples that me and my colleagues have said before, like if we use the right design, the right technique, we would get, of course, the best outcome, or the outcome that we were at least trying to go for it. Like if you're looking for a skin conditions, like creating like a blister model, it would be the right target for you to use it. Like if you want to use the pain model, if you are looking for a drug that it will be impacting on pain, like it would use a pain model. So if you want to look about sleep, like neurophysiology and sleep assessments will be the right thing to implement. And trying to create that value from early stage is it can help a lot, not just for the company, for the drug, but can also save, like in this situation that Teresa was saying, like where we were looking for a food model, like implementing the food model on this study. It helped it massively because we could see not just from the PKs, but of course we have to have that because for the safety of the study, because it's phase 1 study, but having that extra value, like we can see like, oh, there is a biomarker here that we can look forward.
George, I just want to pick up on something there. When you're talking about value in that respect, do you see a lot of additional value having our own sleep labs?
Yes, that's definitely something that I would say like is extremely valuable. Like when if I can say a little bit about that, is like currently it's not just about the sleep lab. I'm gonna be a little bit moving a little bit. But what I want to say is like when the sponsors see like our infrastructure, they not just see the sleep lab. We can see like multiple options that we can use for it. Like one of them like is currently we're doing like psychedelic studies, and we have noticed like in this specific, for example, site where we have in Liverpool, which is CQC accredited, we have the right infrastructures to do the most complex sleep studies. We can go, of course, from an actigraphy watch to actually 24 hours or even more prolonged polysomnographies. And having that infrastructure and the staff with the right expertise is massively helpful. But talking about that structure, I want to say it like, because we have a dedicated room, like where the patient can have like their own in-suite. We have also a dedicated room just for the monitoring, where we can control from the light to the temperature to video monitoring each patient is massively important. Like me coming from the NHS and having, of course, access like to the hospital conditions. Like one of the first things that I noticed when I got to Mac, it was like, wow, this is
no, that's fine. That's
pretty impressive.
Good to hear.
It's like those finer details is what we look for. So we don't just think this is a sleep patient, let's just put them in a room, get them to go to sleep. Is we actually think of how the whole package is and try and reduce as much noise as possible to the study. So small things like if you need to take PKs, so blood samples, and we have little hatches built into the rooms that you can not disturb the patient's experience. So obviously it works really well in psychedelic studies, which we're doing a lot of at the minute. So you put the blood sample in the hatch. You don't have to come in and out of the room. So everything is.
The last thing you want to do is wake someone up.
Exactly. Yeah. There's all these not just implementing that technique. It's actually think about all those finer details and trying to get the data as clean as possible so that it is replicable across all our sites and we can also reproduce it anywhere. There's the same kind of quality of data.
One of the things I've found over the years is that we'll say, oh, yeah, we've got these sleep labs where you could take blood sample through the walls. We've got these models of pain. Companies always come and say, oh, yeah, but what about? Because they know that we've got these access to about 30 million patients via our 10 clinics. They know that we've got the scientists who are developing techniques. What they don't realize is that's what we love. We love it when they come along with a technique or an area we've never worked before, because what we're going to do is make sure that before it goes onto a study for them, we will validate it properly. We're not going to take something on we can't do. It may well be that you want to use a technique which has been developed in academia to a certain level, but we've got to put it into a trial where we could have, last week, the FDA were at one of our units looking at our trials. We could be scrutinized. So we need to make sure we've got the data and the validation to do it properly. And that's the challenge we love. That, we talk about the areas that are developing. We just get excited about that because we want the new areas.
Well, that's perfect. And that's exactly where we need to position ourselves as a business. So if I can just move on, I'm particularly interested in any new techniques that you see coming in. And if so, if there are any specific trends.
I'm sorry, going around the one of the current trends, I think it's undeniable talking about it, is psychedelics. It's been a big thing for Mac over the last 6-7 years. And we have been actually at the forefront of these studies, like being the place where we have done the highest dose, like to the point where talking about the techniques, like we even suggested to the sponsors, like implementing like what we were talking before, like some of these endpoints, some of the additional biomarkers. And one of the things that we are currently working is on EEG while the patient is going through a psychedelic experience. So we can see in real time what is the brain going through. And this is massively because before, when we were discussing with sponsors or other colleagues, all of them said we need to make as less noisy as possible for the patient because we don't want to disturb their experience. But we prove it, we're not just not doing that. We actually, at the forefront, we're already doing that. Not just the EEG, but also evoke potential. So the patient will be even stimulated, with certain sounds.
Absolutely. You can't beat being at the forefront of anything at all.
Yeah.
Teresa.
Yeah, so psychedelics is a massive one as it's coming through clinical research. People will know. So it's definitely a massive area. So we went from, I said, probably 7 years ago, we were one of the leading companies to be involved with this, which is really exciting. And as John said, we love new challenges, and at that time it was new, and it's been in the news about these things coming through. But we managed to go from healthy volunteers into patient populations, and then so the patient populations is looking at sort of depression, anxiety, PTSD, and it's knowing that these are safe, measurable, trained staff, as I do it with our sort of team, will go to the patients rather than patients coming to us. We'll keep that consistency, so we have in-house training as well as the sponsor-specific training for that. But we also test within our team, making sure that we do interoperability testing. And the particular area we test in would be a kappa score, which is how good we are comparing each other. So my scores against George's scores, that is extremely high kappa score. So we have over 90%, which is amazing between rating scales. And then we've gone from that to working with the sponsors of implementing things such as EEGs, which was at the very beginning when those healthy volunteers, it was something that they never thought would be possible because of they didn't know the impacts. And we were able to demonstrate that with sort of having trained people, we were able to implement this and also show not only what is happening from a psychological point of view or a PK point of view, but actually what is going on across the brain. So we can have that data that is...
it's an objective biomarker that we are assessing in real time.
So valuable.
Yeah. And one of the things, the same way we just proved it, we could do this in real time while they're going through this experience. I think one of the next things that we may end up looking is actually about what we spoke it before, about sleep. Because, of course, as Teresa was saying, like we have gone from healthy volunteers to actually conditions like depression, PTSD, all these things, of course, affect their sleep. So if we can now introduce a new objective biomarker like a sleep assessment, it would be massive. Like we're not just proving like from the clinical assessment, the psychological clinical assessment from the rating scales, but we're actually looking as well with the objective biomarker like the sleep is actually affected.
He's sat over there nodding his head. He's chomping at the bit, I think. Not really. Noam, what's your view?
There's two areas where we're getting the new developments coming from. We're keeping a good track on the scientific developments, but what does slightly me is with respect is that a lot of the ideas that people come up with now and are novel are things that we did 30 years ago, and things in our industry do tend to be very phasic. When I first came into the industry, almost every phase 1 study we did had EEG in it. And then people thought, well, no, EEG is not good enough. We've got to go and do these complex imaging studies and brain occupancy studies. And so EEG was pushed away. And now almost every study is once again doing them. So if you wait around long enough, they come around again, these things. For me, it's the challenge is really to keep ahead of what everyone's looking for, what they're developing, try to help people as best we can do. We recognize when we're working with, whether it was a small biotech or one of the huge companies that are out there, you're not actually working with a company, you're working with individuals, and individuals are trying to develop something which is very personal to them. You're taking on almost their baby, if you like, and you've got to treat it with the same respect and thought that they want to. And where I think we're most successful is that when we can actually work directly with the research groups within the pharmaceutical industry, so that we're both working together. It's always been a very open structure at Mac since I worked there. I mean, we've grown in size. I think we're nearly at 400 staff now, but that's because we're now a CRO and we do a lot of work supporting biometrics and bioanalytical labs and things like that. But we still maintain that when we have our regular weekly calls about each of the studies, the sponsor's not speaking to a series of suits, they're speaking to the people who are collecting psychological data on the studies, people who are actually collecting the EEG data, people who are taking the blood samples. They're all gathered together and they've got that contact, and that's really important, I think. And that allows us to be much more flexible in the way we develop. You know, we talked earlier about the work we did in pivoting a sexual function drug into a pain drug. You've got to be that flexible, and you can only make the time and cost savings if you're working very closely with the clients, and that's where I think we work the best.
I totally agree, and it sounds like quite a unique offering in that respect from Mac's point of view. Why don't more companies offer things like that then?
I'm quite happy that they don't, actually.
No.
We quite like
I
being
appreciate we want to
unique.
differentiate ourselves.
It's basically, it's actually very simple. You need three things to make it work. You need great access to patients, which I say is simple, but of course is very complicated. You need validation. You cannot just take an academic technique off the shelf and say, we can do that. It needs to be worked up correctly and have the correct people working on it. You've already alluded to the fact that we have like a very small group of people do each of the testings. We may have 10 centers, but we don't have like 10 physiologists at each site. We move people around who've got the correct experience, so the right people are delivering the study. And that's the final bit. You need the skills. And to bring those together, it's a big investment, both in time and in money. We've made that over the last sort of 20 years the company's existed, and we've got to the point we're at now where it's almost routine for us to do complex patient studies. Yeah, you've got to make the investment earlier. So although perhaps I shouldn't talk about it, other people should be doing it, they're going to have to catch up.
Perfect. Exactly how we want to position ourselves. So I just wanted to ask, well, why don't more sponsors use the techniques that you've referred to in their studies as standard?
I wish more people would do. There's a number of reasons, I think, for that. For me, one of the biggest things which puts off the industry is fear of failure. If you're doing a simple phase 1 study, single ascending dose study with no bells and whistles, the risk is minimal. You may get nothing out of it at the end, but you're not going to have to go to your managers and explain why a pain model failed or why we couldn't measure the anything from a blister that we've created to look for the contents of the fluid. And the reason there is a fear of failure, I think, is because a lot of people have tried these techniques but haven't validated them and run them correctly. Simple example is the cold presser test. If I described it to you, it would sound ridiculously easy. You put your hand in a bath of cold water at 2 degrees centigrade and keep your hand in until it hurts so much that you take your hand out. Very easy technique.
Don't sign me up.
Actually, no, we'll try it with you, Chris. You should try it. Now, in academic centre, I actually took part as a student in an academic centre where I was blindfolded with headphones on and I had my hand in a bath of cold water. And I remember doing it and thinking, oh, this is ridiculous. I can hear people talking behind me. I can hear noise. There was distractions going on. Also, the temperature of the water. I've talked about 2 degrees, very glib. I had my hand in a bucket of water with ice in it. That water would have been somewhere between 1 and 3 degrees. There are pockets of warmer water within it, and you can escape the pain.
Right.
We have a thermostatic control water bath. We have data which will show what happens if the temperature is 1 degree, 2 degree, 3 degree, 5 degree. We know the performance of the test. And so that reduces the variability. Without the validation, there is so much noise that you can't pick out the drug effects. And that's how I think we differentiate, is really with the effort that we put into it in sort of validating it. So that takes a massive effort to do. That's probably one of the reasons why a lot of people will not take this on.
Right. Yeah, that makes a lot of sense. So, finally, and you'll be pleased to know, I just want to ask, can these assessments be conducted anywhere across our global network, or is it just at our MAC-owned sites?
Currently, we are conducting 3 studies using 3 different EEG equipments in multiple sites. And I can guarantee you, because we have a small team and we all have the same in-house training, and even we have to have the training from the sponsors because they want it in a very specific way. Would accommodate that, but what the most important for us is making sure the quality of the data is acquired in exactly the same way, no matter if it's me or if it's Teresa or any other colleague collecting this data from the EEGs or whatever technique we are using. It will have to be with the same standard way. If we have to use a script, for example, for the cold bath, is something that we will implement. For the food model, we'll do the same. So no matter if it's me, Teresa, or anyone else, we'll be doing exactly the same way.
Absolutely. Consistency is absolutely key.
Yeah, it's implementing it. So those finer details, it's not just going to that academic place and putting that model in. We've all gone through academia and have learned sort of a craft from stemming from that, but it's looking at the bigger picture and knowing how do we give the best success as possible for this drug to get onto the market. And it is these finer details so that we can with clarity say to the sponsor that the model that we've implemented has been done with precision, and that we're certain that it sort of is correct and how we've done it. So whatever the outcome is, there's no noise within that data. So it's small details that we add, and from training with our teams, doing iterator reliability testing. So this is not just we get taught how to do it and then go ahead and do it, because there is that tendency of, and they'll know that you can become a bit lax in doing these things. So we'll still test throughout the year. We test within each other and train with each other, buddies with each other. And this can be implemented. This model that we use can be implemented anywhere. We implement it all across our sites. We go to the patients.
But not only can it be used anywhere, we actually do use it anywhere as well, which is quite exciting as well.
Mm-hmm.
As to give out the capsaicin model, for instance, I was asked to run that in about 10 years ago over in Texas. And they wanted to move it to a different center, but the population in San Antonio has a very large Mexican contingent. They were very used to eating chilies, which is where you get your capsaicin from. And so rather than just take the technique over there, we did actually take experimenters over there to do the techniques, so the same people doing it. But we made sure beforehand that people of different ethnic backgrounds who are more used to ingesting the product in a diet would react in the same way when it was injected into the skin. You have to do that level. So a lot of the techniques, although we talk about them around the MAC sites, which is where we tend to run them, we can take on the road, and we do take on the road. I mean, last year, Jordan and I spent a lot of our time on the road doing training for an osteoarthritis study, where we were training both in the patient-reported outcomes in that study, but also giving placebo training, because the biggest problem on pain studies is that people respond to the fact they're getting a treatment, and you get a big placebo effect, and you can try and control that. So in that particular study, we went to over, I think it's like 56 different units in 3 different continents, different countries, different languages. Fortunately, it helps that George speaks more languages than I do. And we trained in how to control the placebo effect. We trained in how the sites should be responding to the patient-reported outcomes, because we can't get our staff to 50 sites around the world. We had to make sure, though, again, to reduce variability, that people were being trained correctly. And this was actually, we actually started that process on another study just before the pandemic hit. And so when we had the problems with COVID in 2020, we had to stop doing that and retreat to a more informal training over Teams. And it was less effective. All of a sudden we saw increases in placebo effect across the studies we were working on, because people don't respond the same way to that intense training that you get. I think one of the funniest things we actually did was we were in a doctor's unit over in, I think it was in Spain, or was it northern Spain? And we gave the talk about placebo and the endpoints, and the guy said afterwards, that was amazing. It was really good. I understand now exactly what we're doing and why we're spending this amount of detail on it. And I said, yes, but you signed off as like a month ago that you did this online training as part of it because we couldn't come because of COVID. And basically we found that really across the board that people were not attending when it was being done over computer. The people don't respond the same way. And so I think what we've learned from that is that we actually make sure that anyone we're working with, any partner sites, wherever they are in the world, and we are working with sites at the moment over in the US and in the UK, on a particular study, that they're all working the same way. Because what we don't want is noise put into the data, and then the study's going to fail, or the study's going to be unclear about the outcomes. We want to make sure we give the best opportunity for a drug to work if it's going to work.
Without doubt. And I think that face-to-face message in that respect is the guaranteed way to get through. And it did, as you say, to get that level of consistency.
I think it's really important.
Wow.
It's quite strange on studies that we did. We went to sites, there was another CRO involved, the sponsor company involved. The only people who went to every single site was the team. Yeah. We saw every site, and they're very, very different.
And
perfect.
And as John said, like sometimes in these situations, like knowing another language is really helpful, because of course before we go in there, like we plan in advance, like we say when we're coming and how we're going to conduct the. And I remember in one of the sites, like they really specify, could you conduct this in our native language? And again, it's helpful on knowing this language.
In like you're actually speaking to somebody in Spanish and Portuguese, and then talking to me in English in the same. I don't know how you do it. Never.
Official translator.
Yes,
that's perfect. Not only have I learnt a lot in that case, but I'd like to thank John, Teresa, and George as far as their time is concerned today. But more importantly, in sharing their knowledge and expertise on how these techniques enable MAC to lead the way in accelerating early clinical development without impacting fundamental need to demonstrate safety, efficacy, and tolerability in a safe and very important regulated environment. By combining our in-house expertise, cutting-edge facilities, and global reach, as John touched upon, we're able to deliver faster, more efficient studies, which is exactly the position we want to put ourselves in. So, just in concluding, I just want to make sure that everyone subscribes to our channel to stay updated on our upcoming episodes of MAC Behind the Trials. And don't forget to follow us on LinkedIn for even more insights. Thank you. And thank you very much to my guests.
Thank you, Chris.